in a limestone cave on the edge of the Dordogne Valley in southwestern France, at a site the excavators call Cussac, a team led by the paleoanthropologist Sasha Kaki lifted the skeleton of a woman from the sediment in 2014 and found something they could not at first explain. Her right humerus, the long bone of the upper arm, carried a ridge of bone along its inner edge nearly twice the thickness of the same ridge in the men buried beside her. She had died approximately 25,000 years ago. She was somewhere between 30 and 40 years old, and the muscle attachment that built that ridge could only have been produced by one motion, repeated tens of thousands of times across her lifetime. The motion of lifting a child to her hip and holding the child there while she walked, while she gathered, while she fled, while she fought. That ridge is called the medial supracondylar crest. It is the anchor point for the brachialis muscle. In the women of Cussac, it had been thickened by a survival pressure so constant, so unrelenting, that her bones remodelled themselves around it. Every adult female skeleton recovered from that cave carried the same signature. The men did not. 25,000 years. 1,000 human generations at 25 years per generation. A stretch of time so long that the glaciers advanced and retreated twice across northern Europe, while these women were lifting children. A stretch of time during which the only thing that stood between a human infant and the predators, the cold, the falls, the rivers, the sudden movements of a herd of bison, was the strength of the woman holding that infant against her body. And I want to ask you something before we go a single step further into this evidence, because the answer is going to shape what comes next. When you carried your children, or your grandchildren, or any child you have ever lifted into your arms, did one side of your body do most of the work? Was there a hip you always reached for first, an arm that took the weight without thinking? I am asking because the women of Cussac chose a side. Their bones tell us which one, and I want to know if your body tells the same story. To understand why this matters, we have to understand what 25,000 years actually means. Chapter 1. The world that made her arms. The world in which the women of Cussac lived was not the world we picture when we hear the word prehistoric. It was not a barren tundra populated by a few scattered figures in fur. It was a complex, layered, demanding landscape that produced one of the most physically capable human populations that has ever existed on this continent, and at the centre of that landscape, doing the work that kept the next generation alive, were the women. 25,000 years ago places us in the middle of what climatologists call the last glacial maximum, the coldest stretch of the most recent ice age. The great ice sheet that covered Scandinavia and the British Isles had pushed its southern edge down into what is now northern Germany. The English Channel did not exist. Britain was a peninsula of the European mainland, and a person could walk from the cliffs of Dover to the mouth of the Thames without ever crossing water. Sea levels were approximately 120 metres lower than they are today. The Mediterranean coastline ran several kilometres further out into what is now open water. The forests of central France were not the oaken beech woodlands we know now, but a vast cold steppe of grass and dwarf birch, dotted with stands of pine in the sheltered valleys, populated by mammoth and woolly rhinoceros, and the great herds of reindeer that moved north and south with the seasons. This is the world the woman at Cussac walked through. To understand her body, we have to walk through it with her. The cave at Cussac was discovered in 2000 by a French cave explorer named Marc Deluc, who crawled through a narrow opening in the limestone and emerged into a chamber whose walls carried engravings of mammoth, bison, horses, and ibex, made by hand with stone tools approximately 25,000 years ago. The chamber was not used for habitation, it was used for burial. 6 human skeletons were eventually recovered from the deeper galleries, placed there with deliberate care, 3 of them women. The dating places them in the cultural period archaeologists call the Gravettian, a tradition that stretched across Europe from approximately 33,000 to 21,000 years before the present. The Gravettian women were not occasional hunters or gentle gatherers in the storybook sense. The isotope analysis of their bones, the chemical signature left in the collagen by what they ate over the last decade of their lives, shows a diet extraordinarily heavy in animal protein. They were eating reindeer, horse, mammoth, fish, and waterfowl. Plant foods made up a much smaller proportion of their calories than in any modern hunter-gatherer population we have studied. They were living, in nutritional terms, much like the wolves and the cave lions, whose remains lie beside theirs in the cave deposits. That kind of diet is only sustained through constant participation in hunting and processing. The work of bringing down a reindeer, butchering it, transporting 40 or 50 kilograms of meat back to camp, scraping the hide, cracking the long bones for marrow. That work did not stop because a woman was carrying an infant. The infant came with her. The work continued. And the body of the woman who did that work for 15, 20, 30 years remodelled itself around the demand. The site at Cussack is not the only place we see this. Approximately 400 kilometres to the northeast, in what is now the Czech Republic, sits a site called Dolní Věstonice. It was excavated beginning in 1924 by the Czech archaeologist Karel Absolon, and the work continues to this day under the direction of researchers from the Moravian Museum in Brno. Dolní Věstonice is older than Cussack by about 3,000 years, dating to approximately 28,000 years before the present, but it belongs to the same Gravettian tradition, and in the female skeletons recovered from Dolní Věstonice, we see the same signature in the upper arm bones. The brachialis attachment is thickened. The deltoid attachment is broader and rougher than in the men. The bones of the forearm, the radius and ulna show curvature consistent with the chronic carrying of weight against the body's centre line. The men of Dolní Věstonice were not weak. Their skeletons show the muscle markings of throwing, the spear-throwing motion that defined the hunt, concentrated in the rotator cuff and the long muscles of the back. But the women's markings were different. The men's arms told the story of release. The women's arms told the story of holding on. This is the first thing I want you to picture clearly. A woman of 28, walking across a cold steppe under a sky that has been grey for weeks. She's wearing layered hide sewn with bone needles that were themselves a Gravettian invention. The eye drilled into the needle so finely that the seam against her skin keeps the wind out. On her right hip, balanced against the iliac crest of her pelvis, is a child of perhaps 18 months. The child weighs approximately 11 kilograms. Her right arm is curled around the child's body, hand cupped under the child's thigh, taking most of the weight. Her left hand is free. In it she carries a digging stick or a basket of plant material or a length of cordage. She has been walking for 2 hours. The child has not been put down. She will not put the child down for another hour and a half, because the ground at this time of year is wet and cold, and there is no surface dry enough to place a small human body on, and because at any moment the herd of horses in the middle distance might bolt, and any child not in arms is a child who cannot be saved. This is the load her brachialis is carrying. This is the load that built the ridge on the inner edge of her humerus. This is what 25,000 years of doing this, generation after generation, did to the female arm. The geography of the Gravettian world made this work even harder. The terrain across which these women moved was not flat. The Dordogne Valley is cut by limestone gorges that drop sharply to the riverbeds and rise again in cliffs that demanded climbing. The plains of Moravia, where Dolní Věstonice sits, are gentler, but they are crossed by rivers that had to be forded in every season, sometimes through ice, sometimes through currents strong enough to sweep an unburdened adult off her feet. A woman crossing such a river with a child on her hip had to lift the child higher, often above her head, while keeping her own balance against the water. The shoulder girdle responsible for that lift, the trapezius, the rhomboids, the levata scapulae, was built by the work itself, and the bone underneath was built to match. The climate added another dimension. At 25,000 years before the present, the average annual temperature in southwestern France was approximately 7 degrees Celsius, colder than it is today. Winters were severe. Snow lay on the ground for months. In such cold, an infant separated from the body heat of an adult could die in less than an hour. The carrying was not optional. The carrying was the difference between a child reaching its second winter and not. Modern thermoregulation studies, conducted by researchers at the Max Planck Institute for Evolutionary Anthropology in Leipzig, have shown that a human infant under the age of 2 cannot maintain its core body temperature in ambient conditions below approximately 15 degrees Celsius without continuous external heat. The most reliable source of that heat was the body of the mother, sister, aunt, or grandmother carrying the child. So the women of Cussack were not carrying children because it was their assigned task in a division of labour. They were carrying children because the alternative was that the children died, and they were carrying for hours, every day of every week of every year, from the time a child was born until the time the child could keep up on foot across long distances, typically around the age of 4 or 5. If a woman bore 4 children across her reproductive life, and the demographic reconstructions from these populations suggest 4 was approximately the average for women who survived to the end of their fertility, then she spent approximately 20 cumulative years of her life with a child in her arms or on her hip, 20 years of bearing weight in a specific posture, 20 years of micro-stress on the same set of muscle attachments. The bone responded. Bone responds to load. This is one of the most well-established principles in skeletal biology, formalized in the 19th century by the German anatomist Julius Wolff, who articulated what is now called Wolff's law, the principle that bone tissue adapts to the mechanical stresses placed upon it. Where load is applied, bone thickens. Where load is removed, bone thins. The astronauts who returned from long stays on the International Space Station lost bone density in their hips and spines because gravity, the most fundamental load on the human skeleton, had been removed. The women of Cussack, conversely, had a load applied so consistently and for so many years that the bone built up additional architecture to handle it. The specific architecture is worth describing carefully, because this is the foundation on which everything else in this script rests. The humerus, the long bone of the upper arm, is not a smooth cylinder. It has ridges, crests, and tuberosities, bumps and ridges where muscles and ligaments attach. When a muscle pulls on bone repeatedly, the bone responds by laying down additional material at the attachment point. Over years, that additional material becomes a visible, measurable ridge. Paleoanthropologists call these enthesial changes, changes at the enthesis, the place where a muscle's tendon meets the bone. In the women of Cussack, the enthesial changes on the medial side of the humerus were graded by the research team using a standardized scoring system developed by the British physical anthropologist Sebastian Villot in 2006. The scoring runs from 0, no observable change, to 3, severe robustness with bone spurs and pitting. The Cussack women scored on average between 2 and 3 on the brachialis attachment. The Cussack men scored on average between 1 and 2. The difference is not subtle. It is the kind of difference that, in modern clinical terms, would mark a manual labourer apart from an office worker. But these were not manual labourers in any modern sense. These were women whose entire physical existence had been organised around one repeated motion, performed thousands of times per week, across decades, across generations, across a stretch of time that spans from before the invention of the bow and arrow to before the invention of agriculture, to before the invention of pottery, to before the domestication of any animal, except, possibly, the dog. And the motion they were performing, the motion that built that ridge, was the motion of protecting a child. There is one more piece of physical context that has to be in place before we begin the central argument, and it is the question of who else was in that landscape with them. The Gravettian women were not alone with the wolves. They lived in social groups that the archaeological evidence at Dolní Věstonice suggests numbered between 20 and 40 individuals, organised in shifting camps that moved with the herds and the seasons. Among those individuals were older women, grandmothers, whose own arm bones recovered from these same sites also showed the brachialis thickening. The lifting did not stop when a woman's own children grew. It continued with her grandchildren, with her sister's children, with any child who needed to be moved across a stretch of ground that was not safe for small feet. This is what 25,000 years of female arm work looked like. Not a single dramatic event, a million small ones, each one repeated by another woman, in another camp, on another cold day, with another child on her hip, while the glaciers advanced or retreated and the herds moved north or south, and the bone in her arm thickened, fibre by fibre, into the shape that the women of Cussack eventually carried into the cave with them when they died. What that shape contained, and what the science has only begun to understand about it in the last 20 years, is what the next 9 chapters are going to lay out, one piece of evidence at a time. Chapter 2. The bone that remembered every lift. The first careful measurement of what protective carrying did to the female arm bone was not made in France. It was made in the early summer of 2017 in a laboratory at the University of Cambridge by a researcher named Alison McIntosh. McIntosh had spent the previous 3 years collecting computed tomography scans of upper arm bones from European female skeletons spanning 7,000 years of prehistory, from the late Mesolithic foragers of approximately 5,500 years before the present, through the early Neolithic farmers of approximately 7,000 years before the present, into the Bronze Age and the Iron Age and beyond. Her sample included 148 female humeri from named excavation sites across Germany, Austria, Hungary and Serbia, and what she was looking for was something no previous researcher had measured in this way. She was measuring the inside of the bone. The interior of a long bone is not solid. It is a hollow shaft called the medullary cavity, surrounded by a layer of dense, compact bone called the cortex, the cortical bone. When a muscle pulls on a bone over many years, the bone responds not only by adding ridges and crests on the outside, but by thickening the cortex itself, by laying down additional dense bone on the inner wall of the shaft. This thickening cannot be seen by looking at a bone from the outside. It can only be measured by cutting the bone in cross-section, or, less destructively, by scanning it with a high-resolution CT scanner that can image the internal architecture without damaging the specimen. McIntosh's scans revealed something that the field had suspected but never quantified. The female humeri across her entire 7,000-year sample showed cortical bone thickness that was, on average, between 9 and 16% greater on the medial, the inner side of the shaft than on the lateral, outer side. This is the side facing the body. This is the side that a child rests against when it is carried on the hip. And this is the side that the brachialis muscle pulls against every time a flexed elbow holds a weight close to the trunk. The asymmetry was not present, or was present only weakly, in the male humeri from the same sites. The men's bones were thicker overall, as is typical for the male skeleton, but the thickening was distributed more evenly around the shaft. The women carried a directional signature, the men did not. This is Revelation 1. The female arm bone across 7,000 years of European prehistory carries a measurable internal architecture shaped by the chronic carrying of weight on the medial side of the body. The architecture is consistent, it is statistically significant, and it appears in every population McIntosh measured. It is not a feature of a single culture. It is a feature of being female in a world where children had to be carried. To understand what this measurement actually means, we have to spend some time inside the structure of the bone itself, because the cortical thickening is only the beginning of what the bone records. The cortical bone of the humeral shaft is made up of microscopic structural units called osteons. Each osteon is a roughly cylindrical column of bone tissue, approximately 200 micrometers in diameter, running parallel to the long axis of the bone. At the centre of each osteon runs a tiny canal, the Haversian canal, that carries the blood vessel feeding that small piece of living tissue. Bone is alive. It is constantly being broken down by cells called osteoclasts and rebuilt by cells called osteoblasts. The process is called remodelling, and it runs continuously throughout the lifetime of every human being. When a section of bone is under chronic mechanical load, the osteoblasts work faster than the osteoclasts, and the cortex thickens. When the load is removed, the balance reverses and the cortex thins. In the women of Mackintosh's sample, the osteons on the medial side of the humeral shaft were not only more numerous than on the lateral side, they were also more tightly packed, more recently remodelled, and oriented along axes of stress that pointed toward the elbow joint. This orientation, measurable through what is called the predominant collagen fibre direction, tells researchers something specific. It tells them which way the load was being applied, and in the female humeri, the load was being applied in the exact direction predicted by the biomechanics of carrying a child on the hip with the elbow flexed at approximately 90 degrees. The bone, in other words, was not just thicker on the side facing the child. It was organised at the microscopic level to resist the precise direction of pull that came from the chronic flexion of the elbow under load. The architecture matched the activity to a degree of specificity that ruled out any general explanation. This was not the bone of women who lifted heavy objects occasionally. This was the bone of women who held a specific weight in a specific posture for an enormous fraction of their adult lives. Mackintosh's findings were published in 2017 in the journal Science Advances under the title Prehistoric Women's Manual Labour exceeded that of athletes through the first 500 years of farming in Central Europe. The title became one of the most discussed phrases in physical anthropology that year, because Mackintosh had done something nobody had done before. She had compared the cross-sectional geometry of her prehistoric female humeri to the humeri of living women, specifically to a sample of female athletes at the University of Cambridge, whose arm bones she scanned for direct comparison. The athletes were rowers, soccer players, and runners, all of them performing at university or national level, all of them training between 6 and 12 hours per week. The bones of the Cambridge rowers, whose sport places extraordinary loads on the upper arms, came close to matching the bones of the Bronze Age women in some measurements, but did not match the women of the early Neolithic. The Neolithic women's humeri exceeded the rowers' humeri in cortical thickness by between 5 and 10%. The strongest arm bones of any group Mackintosh measured, including elite athletes, belonged to women who had been dead for 7,000 years. This finding alone reshaped the way the field thought about prehistoric female labour. But Mackintosh herself, in the discussion section of her paper, noted that the cross-sectional geometry told only part of the story. The pattern of asymmetry, the medial thickening, the orientation of the osteons. These were consistent with one specific activity above all others, an activity that the ethnographic record of every traditional human society documents as primarily female, and an activity that had not been adequately accounted for in any previous study of prehistoric biomechanics. The activity was the carrying of children. Mackintosh's work pointed towards something the field would spend the next decade investigating in detail. If the bone of the female arm had been remodelled by the chronic carrying of infants, then the muscle that did the carrying must have been built to a specification the field had not previously measured. And to measure muscle in a skeleton that has been in the ground for 25,000 years, we have to read the muscle's signature in the bone it was attached to. That signature is what the paleopathologist Sébastien Villot, working at the University of Bordeaux and at the French National Centre for Scientific Research, had spent the previous decade learning to read with new precision. Villot's contribution to this field was the development of a quantitative scoring system for enthesal changes that could be applied consistently across different skeletal collections by different researchers, a methodological advance that allowed the field for the first time to compare findings from sites separated by thousands of kilometres and thousands of years. Villot's system, refined and republished in 2010, identifies 6 fibrous entheses on the upper arm bones that are particularly responsive to chronic loading. 3 of these, the brachialis insertion on the ulna, the biceps brachii insertion on the radial tuberosity, and the common flexor origin on the medial epicondyle of the humerus, are involved directly in flexing the elbow against resistance. The other 3, the deltoid tuberosity, the supraspinatus insertion on the greater tubercle, and the latissimus dorsi insertion on the intertubercular groove, are involved in lifting and stabilizing the upper arm against the trunk. In every prehistoric female skeletal collection that has been scored using Villot's system, the elbow flexion entheses show stronger development than the lifting and stabilizing entheses. In male skeletons from the same sites, the pattern is reversed. The men's bones show the signature of lifting and carrying loads at a distance from the body, the signature of hunting and butchering. The women's bones show the signature of holding loads close to the body, the signature of carrying a child. This is the second piece of physical evidence that Revelation 1 rests on. Across multiple independent sites, scored by independent researchers using a standardized system, the female arm bone shows the enthesial signature of chronic elbow flexion under load in a pattern that is distinct from the male signature and consistent across cultures, climates, and millennia. I want to be careful about one thing here. The bone signature does not prove that every woman in every Gravettian camp spent every waking hour carrying a child. The signature represents an average, a cumulative record of activity across the lifetime of each individual whose skeleton was scored. Some of the women may have carried more, some may have carried less. What the signature does prove is that the activity of carrying weight against the medial side of the body, in a flexed elbow posture, was a sufficiently dominant feature of female life across these populations to leave its mark on the average female skeleton. And the mark is deep. In the Gravettian sample at Dolny Vestonis, the brachialis enthesial scores for the women were on average 80% higher than the corresponding scores for the men. In a comparable sample from the late Mesolithic site of Vlasak, on the Danube River in modern-day Serbia, excavated under the direction of the Yugoslavian archaeologist Dragoslav Srejovic in the 1970s, and reanalysed by Dusan Boric and his colleagues at Cardiff University in the early 2000s, the female brachialis scores ran approximately 70% higher than the male scores. At Çatalhöyük, the great Neolithic settlement in central Anatolia, excavated by James Mellart in the 1960s, and by Ian Hodder and his team from Stanford University from 1993 onward, the same pattern appeared again, slightly less pronounced because the Neolithic women had more time settled in one place, but still clearly distinguishable from the male skeletons of the same site. The pattern is universal across the prehistoric record we have. Wherever the female skeleton has been scored for enthesial changes using a consistent methodology, the elbow flexion entheses are more developed in the women than in the men. What McIntosh's work in 2017 added to this picture was the internal architecture, the cortical thickening, the osteon orientation, the directional signature inside the bone, that confirmed what the enthesial signature on the outside of the bone suggested. The women's arms had not just been used, they had been remodelled. The bone itself had changed shape to accommodate a load it expected to keep carrying. The question that this evidence raises, the question that the second chapter of this script has set up but cannot yet answer, is this. If the bone changed shape across the lifetime of each individual woman, and if the change is visible in skeletons across 25,000 years and across 2 continents, then something in the female body was responding to the carrying load in a way that the male body was not. The men of these populations carried things too. They carried weapons, they carried meat, they carried each other when injured, but the men's arm bones do not show the same medial signature. Why? The answer to that question is going to require us to step away from the bone for a moment and look at what was happening inside the muscle itself. Because the muscle, it turns out, was being built by something more than mechanical load alone. The muscle was being built by a chemistry that the female body had developed specifically for this work, a chemistry that the next chapter is going to lay out, with one finding that complicates the simple story we have just begun to tell. Chapter 3. The chemistry that built the grip, in 2019, 2 years after McIntosh published her cortical bone findings, a research team at the University of Oklahoma, led by the human evolutionary biologist Kara Okobock, began publishing a series of papers that approached the question of female muscular adaptation from a completely different direction. Okobock's team was not looking at bones. They were looking at living muscle in living women, and they were measuring something that the skeleton alone could not reveal, the chemistry of how the female muscle is built and how it responds to load. What they found was the first complication in the story we have been telling, and it is the kind of complication that holds an audience through the middle of a long inquiry, because it does not contradict the bone evidence, it deepens it. The female skeletal muscle, Uckerbock's team confirmed and extended what a number of earlier studies had begun to suggest, is built from a different fibre type composition than the male skeletal muscle. Human skeletal muscle is made up of two broad categories of muscle fibres, traditionally called Type 1 and Type 2. Type Y fibres, also called slow twitch fibres, contract relatively slowly but resist fatigue extraordinarily well. They are densely packed with mitochondria, the small structures inside each cell that produce energy through the burning of oxygen, and they are the fibres that allow a muscle to sustain a continuous, moderate load for hours without giving out. Type 2 fibres, fast twitch fibres, contract quickly and powerfully but fatigue rapidly. They are the fibres that produce explosive movements like sprinting or throwing. The female body, on average, has a higher proportion of type 1 fibres in nearly every muscle group that has been measured. In the brachialis, the muscle that does the work of holding a flexed elbow under load, the female type 1 proportion runs approximately 10 to 15% higher than the male proportion. In the soleus, the deep calf muscle that maintains posture during walking and standing, the difference is larger, running closer to 20%. In the trapezius, the muscle of the upper back that stabilizes the shoulder girdle when an arm is holding a weight at chest height, the female type 1 dominance is again pronounced. This is revelation 2. The female muscular system is built, at the level of its fundamental fibre composition, for sustained moderate load rather than for brief explosive output. The fibres that hold a child for 3 hours of walking are not the same fibres that throw a spear at a fleeing reindeer. The female body has more of the former, the male body has more of the latter, and the difference is present across populations, across diets, across activity levels, and across age groups. The complication that Uckerbock's team introduced, and this is what makes the third chapter of this script the place where the simple story stops being simple, is that the female muscle does not just have more type 1 fibres, it also recovers from fatigue faster than the male muscle, and it uses fat as a fuel more efficiently than the male muscle does. A 2010 study from the University of British Columbia, conducted by the kinesiologist Brian Dalton and colleagues, measured the time it took for the same muscles in matched groups of men and women to recover their full, force-producing capacity after a standardized fatigue protocol. The men, who produced higher peak forces during the initial contractions, took on average between 15 and 20% longer to return to baseline than the women did. The women's muscles, despite producing lower peak forces, came back faster. They were ready to work again sooner. A separate body of research, much of it conducted by the Canadian exercise physiologist Mark Tarnopolsky over the course of the 1990s and 2000s, established that women, during sustained moderate exercise, derive a larger fraction of their energy from the oxidation of fatty acids, the burning of stored body fat, than men do under identical conditions. This is not a small effect. It runs between 5 and 12% depending on the conditions of the test. And it has direct consequences for what the body can do over long periods. The combination is what matters. A higher proportion of slow twitch fibres, faster recovery between bouts of effort, and greater reliance on fat as fuel during sustained activity. Each of these adaptations on its own would make the female body slightly better suited to long, continuous, moderate intensity work. Together, they make the female body extraordinarily suited to one specific activity above all others, the activity of holding a moderate weight close to the body, walking with that weight for hours, occasionally adjusting position, or absorbing a sudden shift in the load, and continuing this work day after day after day without collapse. This is the chemistry that built the grip the bone of Cussack recorded. The bone tells us what the muscle was doing. The muscle tells us how it was able to do it. Now I want to pause here, because the implication of what I just laid out is something the audience watching this video has lived inside her own body, without ever having it named. And I want to know whether the description is going to land. Have you noticed, in your own life, that the kind of physical work that wears out the men around you, the lifting, the holding, the kind of task that requires patience as much as power, is the kind of work you find that you can do for far longer than they can? Not faster, not with more sudden force, but for longer, without stopping, without the same depletion. I am asking because the bone evidence and the muscle evidence are converging on a picture of the female body that says exactly this, and I am curious whether you recognise yourself inside it. The deeper picture that the field has been building since approximately 2015 is that the female body's metabolic preference for fat oxidation during sustained activity is mediated by a specific set of hormonal mechanisms that the next chapter is going to address in detail, and that these mechanisms produce different muscle behaviour in women than in men, even when the two groups are matched on training, body composition, and age. But before we get to the hormones, I want to give you the second piece of evidence that the muscle chemistry conclusion rests on, because this is where the third chapter introduces its specific complication. The evidence comes from the analysis of mitochondrial DNA in skeletal muscle samples taken from living women of different ages. A research group at McMaster University in Ontario, led by the muscle biologist Gianni Parise, published a paper in 2014 examining the mitochondrial content and function of muscle biopsies taken from the vastus lateralis, a major thigh muscle, in men and women across an age range from 20 to 70 years. The team found that women's muscle mitochondria, at every age studied, were on average more densely packed and more metabolically active than men's mitochondria from the same muscle, measured on a per-fibre basis. The difference was present in 20-year-olds. It was present in 70-year-olds. And while both sexes lost mitochondrial density with age, the women started higher and ended higher. Mitochondria are the structures inside each muscle cell that produce energy. More mitochondria, packed more densely, with higher activity means more energy production per unit of muscle. And critically, more mitochondria mean more capacity to burn fat for fuel during sustained activity, because the fatty acid oxidation pathway runs through the mitochondrial membrane. This is the chemistry that supports the muscle that supports the bone. The female body, from the cellular level up through the muscle fibre composition up through the bone architecture, is organised for sustained, moderate, continuous load. The complication, and this is what makes the third chapter's evidence harder to interpret than the second chapter's, is that the same chemistry that supports sustained carrying also imposes a cost. The female body, organised for endurance work, is not organised for explosive power. The peak force a female brachialis can produce in a single maximum contraction is, on average, between 30 and 40% lower than the peak force a male brachialis can produce. This is not because the female muscle is weaker per unit of cross-sectional area. When force is measured per square centimetre of muscle, the difference is much smaller, on the order of 5 to 10%. The difference is because the female brachialis is, on average, physically smaller than the male brachialis. So we have a paradox in the bone record. The female humerus shows medial thickening that, by Wolff's law, can only have been produced by chronic load. The female muscle is organised for sustained moderate work, but the female muscle is also smaller, on average, than the male muscle. How does a smaller muscle, working through a smaller bone attachment area, produce the level of cortical remodelling that McIntosh measured? The answer, and this is the third piece of evidence in this chapter, is that the load on the female arm in a child-carrying posture is not a peak load. It is a sustained moderate load applied for an enormous number of hours. The cumulative mechanical signal to the bone, the integral of force times duration, is what drives the remodelling, not the peak force. And the female body's muscular chemistry is specifically organised to deliver that kind of cumulative signal. A 2013 biomechanical study at the University of California, Berkeley, led by the anthropologist Erin Wallace, measured the load on the brachialis and on the entire flexor compartment of the upper arm during simulated infant carrying. The study used adult women carrying weighted infant dummies on their hips for sustained periods, with surface electromyography measuring muscle activation throughout. Wallace's team found that the average activation of the brachialis during hip carrying ran at approximately 15 to 20% of maximum voluntary contraction, a moderate level. But the activation was continuous. There was almost no off-cycle, no period during the carrying when the muscle was allowed to fully relax. The shoulder might shift, the grip might adjust, but the brachialis was holding the weight without interruption. This is the load profile that builds bone. Not the brief maximum effort of lifting a stone, but the long, continuous, never-released contraction that holds a child against the body for hour after hour after hour. And the chemistry of the female muscle, the slow twitch of fibre dominance, the dense mitochondrial packing, the efficient fat oxidation, the rapid recovery, is the chemistry that makes that load profile sustainable. A muscle dominated by fast-twitch fibres, low in mitochondria, dependent on glycogen for fuel, could not hold that load for those hours. It would fatigue, the carrying would fail, and the child would fall. The female muscle is the muscle that does not fail under that load. This is what Revelation 2 means. The female body's muscular chemistry is not a generic version of the male body's muscular chemistry, adjusted downward in scale. It is a different chemistry, organised around a different load profile, optimised for a different kind of work. The work it is optimised for is the work of holding small humans against the body for as long as those small humans need to be held. The complication is that this optimisation comes at the cost of peak power. The female body trades the ability to throw a spear hard for the ability to carry a child long. The trade was not, in evolutionary terms, a sacrifice. It was an investment. The species needed both kinds of work to be done. The male body was organised for the explosive work. The female body was organised for the sustained work. The species survived because both organisations existed in complementary form within the same population. But the female version of that organisation had its own internal pressures, its own internal signals, its own internal chemistry that the bone alone cannot reveal. The hormones that drive female muscle development, the hormones that determine which fibres grow and which retreat, which mitochondria proliferate and which die back, which entheses thicken and which remain quiet, were responding to a survival pressure that the next chapter is going to address. The pressure was constant, the hormones were specific, and the result by the time the women of Cossack were buried in their cave was a body whose every layer, from the bone outward, had been organised around the work of protecting children. That hormonal mechanism, and the way the female body uses it to amplify the muscular response to chronic carrying, is what Chapter 4 will lay out, beginning with a single finding from a single research team that changed the way the field thought about how women build muscle. Chapter 4: The Hormones That Kept Her Holding. In 2008, a research team at McMaster University in Ontario published a paper in the American Journal of Clinical Nutrition that at the time attracted very little attention outside the narrow world of muscle physiology. The lead author was a young researcher named Stuart Phillips, working under the mentorship of the same Mark Tarnopolsky, whose earlier work on female fat oxidation we have already discussed. The paper measured the rate at which muscle protein was being built, in real time, in the legs of men and women after a bout of resistance exercise. The technique Phillips and his team used is one of the most powerful tools modern muscle biology has developed. It involves the infusion of a tagged version of an amino acid, in this case, the amino acid phenylalanine, marked with a stable isotope of carbon, into the bloodstream of the research participants. The tagged amino acid circulates through the body and is incorporated into newly synthesized muscle protein at the same rate as the untagged version. By taking small muscle biopsies at specific time points after exercise, and by measuring the ratio of tagged to untagged phenylalanine in the muscle protein, researchers can calculate the fractional synthesis rate. the percentage of muscle protein being built per hour. What Phillips' team found in 2008 was that the fractional muscle protein synthesis rate, measured in this way, was approximately the same in men and women in the hours immediately following resistance exercise. The female muscle, gram for gram, was building protein at the same rate as the male muscle. The difference between male and female muscular development, the team concluded, was not a difference in the rate at which protein was being assembled. The difference was somewhere else. This finding, modest as it sounded, opened a door. If women were synthesizing muscle protein at the same rate as men, then the smaller average size of female muscle had to be explained by something other than synthesis. It had to be explained by either lower starting muscle mass, different patterns of activation, different signals telling the muscle when and where to grow, or some combination of these factors. The work that followed, over the next decade, identified the signals, and the signals were hormonal, and the hormones turned out to be more complicated than the field had assumed. Revelation 3, the central mechanistic finding of this chapter, is that the female body uses a specific hormonal signalling architecture to build and maintain the muscle of chronic carrying, and that this architecture is dominated not by testosterone, the hormone most commonly associated with muscle growth, but by a combination of estrogen, growth hormone, and a less familiar hormone called insulin-like growth factor 1, often abbreviated IGF-1, a hormone produced primarily in the liver and in muscle tissue itself, which signals the body to build and maintain muscle protein. The traditional understanding of muscle growth, dominant in exercise science for decades, was that testosterone, the primary androgen, the hormone produced in large quantities in the male body and in smaller quantities in the female body, was the central driver of muscle protein accretion. This view was supported by the observation that men, with circulating testosterone levels approximately 15 times higher than women's, developed larger muscles in response to identical training stimuli. The conclusion seemed obvious. Testosterone was the muscle hormone. Women had less of it. Women had less muscle. But the conclusion turned out to be incomplete, because the female body, while producing very little testosterone, produces other hormones that drive muscle development through different pathways. And these other hormones, in the conditions that prehistoric women lived in, sustained moderate load, abundant protein in the diet, adequate body fat stores, regular cycles of reproductive activity, are extraordinarily effective at building and maintaining the specific kind of muscle that the female body uses for sustained work. Estrogen, the primary female sex hormone, has been understood since the 1990s to play a direct role in muscle maintenance. The work of the Italian researcher Marco Sandri at the University of Padova and the work of the American physiologist Dawn Lowe at the University of Minnesota established through the 2000s and 2010s that estrogen receptors are present on skeletal muscle fibers, and that estrogen acts on these receptors to suppress muscle protein breakdown. The hormone does not primarily drive synthesis. It blocks degradation. It tells the muscle, in chemical terms, do not take this fiber apart. It is still doing useful work. Lowe's lab published a sequence of papers between 2010 and 2017 demonstrating that the loss of estrogen, either surgically through the removal of the ovaries in animal models or naturally through menopause in human studies, was followed by a measurable decline in muscle mass and muscle force production. The decline was reversible in animal models by the administration of estrogen replacement. The decline was not reversible in humans in the same complete way, because human menopause involves more than just the loss of estrogen, but the directional finding was clear. Estrogen protects muscle. When estrogen falls, muscle is lost faster than it can be rebuilt. This is the first piece of the hormonal puzzle. The female body, for the entire span of reproductive life, typically from the early teens through the late 40s, maintains a hormonal environment in which estrogen is continuously preserving muscle tissue from degradation. The same muscle, in the same activity profile, in the same nutritional environment, will be larger and more durable in a premenopausal woman than in a postmenopausal woman. because the hormonal environment is doing different work in each case. For the women of Cossack, of Dolny Vestonis, of every Gravettian camp across Europe, this hormonal preservation effect was active for the entire span of their childbearing years, which was the entire span during which they were doing the carrying. The estrogen was building the chemical environment in which the muscle could absorb the chronic load without breaking down. The second piece of the hormonal puzzle is growth hormone. Growth hormone is produced in the pituitary gland, a small structure at the base of the brain, and it is released in pulses throughout the day, with the largest pulse typically occurring in the first hours of deep sleep. Growth hormone signals the liver to produce insulin-like growth factor 1, which then circulates throughout the body and binds to receptors on muscle, bone, and other tissues, driving the building of new protein. What the work of researchers including Anne Friedlander at Stanford University and Jill Canale at the University of Missouri established through the 2000s and early 2010s is that the female pattern of growth hormone secretion is distinctly different from the male pattern. Women release growth hormone in higher peak pulses, more frequently throughout the day, and with a different responsiveness to physical exertion. The female pituitary, in essence, is more reactive to the signal of sustained moderate work than the male pituitary is. A long walk with a child on the hip is, hormonally, a more potent stimulus for growth hormone release in a woman than the same walk would be in a man. This means that the very activity that was loading the female brachialis, the chronic carrying of a child on the hip, was also driving the hormonal signal that built the brachialis. The work created its own anabolic environment. The muscle was being told to grow by the same activity that was creating the demand for it to grow. The third piece of the hormonal puzzle is insulin-like growth factor 1, the messenger that growth hormone uses to do most of its work in muscle. IGF-1 is produced both in the liver, in response to circulating growth hormone, and locally in the muscle itself, in response to mechanical stretch and contraction. The locally produced IGF-1, often called mechanogrowth factor when it is generated by physical loading, is what drives the specific targeted growth of the muscles that are being used. When the female brachialis was contracting continuously to hold an 11-kilogram child against the body, the muscle fibers were releasing mechanogrowth factor at the site of the contraction. The growth factor was binding to receptors on the same fibers. The fibers were receiving the signal to synthesize new contractile protein. The bone, in turn, was receiving signals from the loaded muscle that triggered the osteoblasts to lay down additional cortical bone at the attachment point. This is the mechanism that explains, at the cellular level, what McIntosh and Villot had measured at the skeletal level. The chronic loading of the female arm was driving a local hormonal cascade that built the muscle, the enthesial attachment, and the cortical bone all in response to the same activity, all in the same direction, all in the same anatomical location. And here is the additional finding that this chapter extends the mechanism with, because the story does not end with the local cascade. The estrogen environment of the female body amplifies the IGF-1 response in muscle. Studies by the Danish physiologist Michael Kjær at the University of Copenhagen, conducted through the 2010s, demonstrated that estrogen sensitizes muscle tissue to the anabolic effects of IGF-1. The same dose of IGF-1 in a muscle exposed to physiological estrogen produces more protein synthesis than in a muscle without estrogen exposure. So the full mechanism, as it is now understood, runs like this. A woman in her reproductive years walks for 2 hours with a child on her hip. The brachialis fires continuously to hold the child. The pituitary releases growth hormone in response to the sustained moderate exertion. The liver produces IGF-1. The brachialis releases additional, locally produced IGF-1 at the site of contraction. The IGF-1 binds to receptors on the brachialis fibers. The estrogen circulating through her body sensitizes those receptors. The protein synthesis machinery inside the muscle cell is activated. New contractile protein is built. New attachment fibers are laid down at the enthesis. New cortical bone is deposited beneath the enthesis. The arm becomes more capable of carrying the next child she lifts. This is the chemistry that the prehistoric female arm was running on. Not testosterone, which the female body produces in small amounts, estrogen, growth hormone, and IGF-1, working together, building the specific architecture that the work of carrying required. The implication of this mechanism, and the additional finding that extends it beyond what the field had assumed even 10 years ago, is that the female body's response to chronic moderate loading is, in some respects, more efficient at building and maintaining the specific muscle of sustained work than the male body's response would be to the same loading. The male body, with its dominant testosterone signaling, builds larger peak force capacity, but does not build the sustained load architecture as efficiently. The female body, with its estrogen IGF-1 architecture, builds smaller peak force, but a more durable, more recoverable, more sustained work-capable muscle. The species, in evolutionary terms, had access to both kinds of bodies. The work that required peak force was done by the male body. The work that required sustained capacity, and the most fundamental sustained capacity work of all was the carrying and protecting of children, was done by the female body. And the hormonal architecture of the female body had been organised across hundreds of thousands of years of selection pressure to do that work better than any other configuration of the human form could have done it. That architecture, that hormonal signature, that integrated mechanism running from the pituitary, through the bloodstream, through the muscle fibre, through the enthesial attachment and into the cortex of the bone, is what the women of Cussack were running on the day they died and were carried into the cave by their kin. It is what the women of Dolny Vestonis were running on, and it is what every woman who has ever picked up a child has been running on, whether she knew the names of the hormones or not. The question this chapter has set up but cannot yet fully answer is whether the same architecture appears in populations completely outside the European prehistoric record. If it does, if a woman buried in a Neolithic grave in Anatolia, or in a Mesolithic grave in the Levant, or in a hunter-gatherer settlement in the high steppe of Central Asia, shows the same hormonal skeletal signature, then we are looking at a feature of the female body that is not local. We are looking at a feature that selection pressure built into the entire species, across continents and across time, because the work of protecting children was the same work everywhere humans lived. That is the question Chapter 5 is going to address, by travelling away from the limestone caves of southwestern Europe and looking at what the female skeleton looks like in the cemeteries of 3 other ancient worlds. Chapter 5. The Evidence from 3 Worlds. The first of these other worlds is at the eastern end of the Mediterranean, in a part of the Levant that today straddles the modern borders of Israel, the Palestinian territories, Jordan, Lebanon and Syria. Approximately 15,000 years ago, the people who lived in this region began doing something that no human population had done in any previous era. They began staying in one place. They built villages of round stone houses, half buried in the ground for insulation, roofed with thatch. They harvested wild grain with sickles made of bone hafted with flint blades. They buried their dead beneath the floors of their houses. The cultural tradition they belong to is called the Natufian, and it is one of the most carefully studied transitional populations in all of archaeology, because the Natufian stood at the beginning of the human shift, away from full mobility and towards settled life. The Natufian women were not Gravetian women. They lived in a warmer climate. They did not face the same long winter snows. Their diet was more plant-based, drawing heavily on wild barley, wild emmer wheat, almonds, pistachios, and gazelle. Their houses gave their children a place to be set down, sometimes in a way the open camps of the Gravetian could not. And yet, when their skeletons are examined, the female arm bones from Natufian cemeteries carry the same signature that the European women's bones carried. The medial thickening of the humerus, the strong brachialis enthesis, the cortical asymmetry favouring the inner side of the shaft. The work of documenting this in the Natufian record was carried out primarily by the Israeli paleoanthropologist Israel Hershkovitz at Tel Aviv University, with collaboration from researchers at the Hebrew University of Jerusalem and the French Centre National de la Recherche Scientifique. Hershkowitz's team examined the skeletal collections from Natufian cemeteries at Einan, also known as Ain Malaha, in the upper Jordan Valley, and at El Wad Cave on the western slope of Mount Carmel, and at Hayanim Cave in the western Galilee. Across these three sites, spanning approximately 3,000 years of Natufian occupation, the female Humeri showed the same pattern that Mackintosh would later document in the European sequence. The medial side of the shaft was thicker. The brachialis attachment was more developed in the women than in the men. The asymmetry was consistent across the population. This is Revelation 4. The carrying signature in the female skeleton is not a feature of one culture, one climate, or one continent. It appears in populations separated by thousands of kilometres, in environments that demanded different specific patterns of daily activity, in cultures whose material technologies and subsistence strategies bore little resemblance to each other. The signature is a feature of being female in a world where small humans had to be carried by adult ones, and that world was, for the entire span of prehistory, every world there was. The second of the other worlds I want to take you to is much further east, on the steppe of Central Asia, in a region that today straddles southern Russia, northern Kazakhstan, and western Mongolia. Approximately 5,000 years ago, the populations of this region were undergoing one of the great transformations in human history. They were domesticating the horse, they were learning to ride, and the women among them, whose skeletons we now have access to through the work of a number of Russian and Kazakh excavation teams, carried the carrying signature in their arm bones, alongside an additional set of adaptations that the horse imposed. The site I want to name specifically is Botai, in northern Kazakhstan, excavated initially in the 1980s by the Kazakh archaeologist Viktor Zybert, and more recently reanalysed by an international team led by Alan Outram at the University of Exeter. The Botai people were not, in the strict sense, riders in the way later steppe populations would be. They were a transitional population, beginning to domesticate the horse for milk, for meat, and possibly for limited mounted herding. The female skeletons from Botai show the medial humeral thickening that we have now seen in Cusack, in Dolny Vestonis, in the Natufian cemeteries of the Levant. But they show something else as well. They show signs of horse-related activity in the lower limb, in the femur and the pelvis, and they show wear patterns on the cervical vertebrae of the neck consistent with carrying loads on the head, a posture that women in many traditional societies have adopted for water and grain transport. The combination is what interests me here. The Botai women were doing horsework and head-carrying work and child-carrying work, and the bones record all three. The carrying of children in particular had not been displaced by the addition of horses to the economy. It had simply become one more category of physical labour that the female body was expected to absorb. And the brachialis signature in the Botai women's humeri runs as strong as it does in the European prehistoric populations, perhaps slightly stronger, because the Botai women were carrying children across longer distances on more varied terrain than their European contemporaries. The work of measuring this in detail was published in 2019 by a research team that included specialists from the Russian Academy of Sciences in Novosibirsk and the German Archaeological Institute in Berlin. The team's lead author, Svetlana Sharapova, working with the bioarchaeologist Alexandra Buzilova, documented the specific cross-sectional geometry of the female humeri at Botai using the same methodology Mackintosh had developed for the European samples. The findings matched. The female humeri showed thicker cortical bone on the medial side. The brachialis attachment was strongly developed. The asymmetry was statistically significant against the male sample from the same site. The third world I want to take you to is the furthest from Europe in cultural terms, and in some respects the most surprising in what its skeletons reveal. It lies in what is now southwestern Mexico, in the highland valleys of Oaxaca, where a long sequence of human occupation runs from approximately 10,000 years before the present through the rise of the Zapotec civilization at Monte Alban, approximately 2,500 years ago. The skeletal collections from this region have been studied extensively, most prominently by the American physical anthropologist Lourdes Marquez Morfin, working at the National Institute of Anthropology and History in Mexico City, and by her collaborators at the Universidad Nacional Autónoma de Mexico. The Oaxaca Valley women carried children across a different landscape than any we have looked at so far. The valley is high, approximately 1,500 metres above sea level, and the terrain rises sharply on every side into mountains that reach over 3,000 metres. Daily life involved climbing, the work of gathering, of fetching water, of moving between fields and villages, required ascending and descending steep slopes. A woman carrying a child up such a slope is not just holding the child against her body. She's supporting the child's weight against the additional load of her own body's vertical motion. The brachialis is contracting harder. The shoulder girdle is stabilizing against larger forces. The bone, by Wolff's law, should be responding correspondingly. It is. Marquez Morfin's published analyses of the Oaxaca female skeletons, drawing on collections from sites including Yagul, Mitla, and Monte Alban itself, document the same medial humeral thickening that the European and Asian populations show. The signature is, if anything, slightly more pronounced, which is consistent with the additional mechanical demand of vertical terrain. The brachialis emphasis is large. The cortex on the medial side runs visibly thicker. The asymmetry is, again, statistically robust against the male sample. 3 worlds, 3 independent skeletal records, 3 different climates, 3 different diets, 3 different cultural traditions, all 3 carrying the same signature in the female arm. This is what Revelation 4 is for. The argument we are building cannot be a local one. If the medial humeral thickening were a feature of one population, it could be explained by anything, by a peculiarity of the diet, by a particular cultural practice, by the geography of one valley. But when it appears in Cussack and in Hainan and in Botay and in Monte Alban, in populations separated by tens of thousands of kilometres and tens of thousands of years, the explanation has to be deeper than any local factor. The explanation has to lie in the female body itself, in a feature of female biology that responds to the universal demand of protecting children with the same skeletal architecture wherever that demand is imposed. That feature, the field has converged on understanding, is the integrated hormonal muscular skeletal mechanism we laid out in Chapter 4. The mechanism is not cultural, it is not regional. It is built into the female human body by selection pressure that operated on every population for the entire span of human evolutionary history. The pressure was the same everywhere, because children were the same everywhere. Small, vulnerable, slow to grow, requiring constant physical contact with an adult for protection during the years when they could not protect themselves. And the response of the female body to that pressure was the same everywhere. The brachialis grew, the bone thickened, the cortex remodeled, the hormones organized themselves around the work, the muscle chemistry shifted towards sustained load. The skeletal record carries the signature in every prehistoric female population that has been examined with the methods to detect it. I want to add one specific piece of evidence to this chapter, drawn from a researcher we have not yet named, because the addition deepens the picture in a way that matters for what comes next. The researcher is Daniel Westcott, an American physical anthropologist working at Texas State University, who in 2006 published a comparative study of cross-sectional geometry in human humeri from 23 different population samples, ranging across modern and ancient groups, hunter-gatherer and agricultural, Old World and New World. Westcott's analysis demonstrated that the male-female difference in humeral cross-sectional shape is one of the most robust sex differences in the human skeleton, more robust, in some respects, than the differences in the pelvis that are traditionally used to determine sex from skeletal remains. The female humerus is more asymmetric, the male humerus is more symmetric, and the direction of the female asymmetry across every population Westcott measured points toward the medial side of the shaft. This finding extends the geographic and chronological range of the carrying signature even further than the 4 sites I've walked you through. Westcott's sample included populations from across Africa, from Native American groups in North and South America, from Australian Aboriginal collections, from East Asian historical samples, from Pacific Islander populations, and from European medieval cemeteries. The carrying signature appeared in every one of them. The female body, wherever it has lived, has carried children. The bones of the female arm record that carrying, and the chemistry, the architecture, the integrated mechanism that produced those bones is the same in Oaxaca as it is in the Dordogne, the same in the Levant as it is in Kazakhstan, the same in Pacific Islander populations as it is in medieval European villages. This is what 25,000 years built into the female body, not into one woman, not into one population, into the species in its female form wherever the species existed. The question we are now ready to ask is one that the bone evidence cannot fully answer on its own. If this architecture was built by chronic carrying across hundreds of thousands of years, and if it is present in every prehistoric female population we have examined, then what does this architecture do in a body that is no longer required to carry children for hours every day? What happens to the brachialis, to the hormonal cascade, to the cortical bone, in a female body whose work life looks nothing like the life of the women at Cussack? The answer to that question is the most counterintuitive finding in this entire script, and it is what Chapter 6 is going to address. Chapter 6. The finding nobody expected, when researchers began in the 1990s to apply the methods of paleopathology to the skeletal collections of modern industrialized populations, when they began to measure cortical thickness and enthesial development in the bones of women who had lived in cities, who had worked in offices, who had raised children in apartments with washing machines and strollers, they expected to find something straightforward. They expected to find that the carrying signature, the medial humeral thickening that was so pronounced in the Gravettian and the Natufian and the Botai and the Oaxaca women, had begun to disappear. They expected, in essence, that the disuse of the female arm for sustained child carrying across the few generations of industrial and post-industrial life would have produced exactly what Wolff's law would predict. The load was gone. The cortex should be thinning. The enthesis should be smaller. The architecture should be unwinding. What they found instead is what makes the 6th chapter of this script the place where the simple story does not just become complicated. It becomes something different from what the field had expected. Revelation 5, the counterintuitive finding that this chapter rests on, is that the carrying signature in the female arm has not significantly diminished across the populations of the modern industrialized world. It has changed in some ways. It has weakened in some specific subgroups. But it has not disappeared, and in some populations it has even strengthened, because the female body is finding new ways to express the same architecture in response to new loading patterns that researchers had not initially recognized as functionally equivalent to the carrying of children. The first careful documentation of this came from a comparative study published in 2014 by a research team at the University of Tübingen in Germany, led by the bioarchaeologist Joachim Wahl. Wahl's team measured cortical bone thickness and enthesial development in female humeri from 3 populations: a medieval German cemetery dating to the 12th century, a 19th century industrial-era German cemetery, and a sample of modern post-mortem specimens from the same region. The expectation was a smooth downward gradient across the 3 samples. The medieval women would show the strongest carrying signature. The industrial women would show a weaker version. The modern women would show the weakest version. The result was not a smooth gradient. The medieval women showed strong carrying signatures, as expected. The industrial women showed somewhat weaker signatures, as expected. But the modern women, the contemporary sample, did not show the dramatic reduction the team had anticipated. The cortical thickness on the medial side of the humerus remained more developed than on the lateral side. The brachialis enthesis remained more developed in the women than in the men of the same modern sample. The architecture, attenuated but recognizable, persisted. Why? The team's analysis, which they extended in subsequent papers through the late 2010s, pointed to several factors that together account for the persistence. Modern women, even in highly industrialized societies, still carry children. They carry them on the hip during the years of infancy. They carry them in front pack carriers. They lift them in and out of car seats, strollers, baths, beds. The cumulative carrying time per individual mother is lower than it was in the Gravettian, but it is not zero, and the female body responds to even attenuated chronic loading with the same architectural signature that it expressed at full intensity in the prehistoric era. But the persistence is not only about residual child carrying, it is also about something the field had not initially considered as functionally equivalent to carrying, and this is where the counterintuitive finding becomes truly counterintuitive. The modern female body has found new chronic moderate loading patterns that activate the same hormonal muscular skeletal cascade that the carrying of children activates. The carrying of grocery bags, often in the same medial side posture that infants are carried in. The chronic supporting of weight on the hip in a wide range of household tasks. The carrying of laundry, of pets, of children's gear, of the materials of daily life, and in a finding that has emerged most clearly in the last decade, the carrying of the weight of one's own body across long periods of standing in occupations that the modern female workforce occupies in disproportionate numbers: nursing, teaching, retail, caregiving, food service. The work of the British biomechanist Alison McGregor at Imperial College London, published in a series of papers between 2015 and 2021, has begun to map the load profiles of these modern occupations against the load profiles of prehistoric child carrying. The findings are striking. A nurse working a 12-hour shift, repeatedly lifting and supporting adult patients, undergoes a cumulative loading profile on the upper arm that resembles, in its duration and its sustained moderate intensity, the loading profile of a Gravettian mother carrying an infant on her hip across a day's gathering. The peak forces are different, the specific postures are different, but the integrated cumulative load, force times duration, falls in a comparable range. This is the second piece of evidence in the chapter, and it is what extends Revelation 5 into territory the field is still actively investigating. The female body's chronic loading architecture has not become obsolete in modern life. It has been redirected. The same brachialis, the same medial cortical thickening, the same hormonal cascade that built the arm of the woman at Cussack is being expressed in modern female bodies whose daily loading is no longer dominated by infants, but is still dominated by sustained moderate physical demand of one form or another. And here is where the finding gets sharper. In populations of modern women whose lives genuinely do not include chronic moderate loading, sedentary office workers in highly automated environments, with no children or with adult children, with no significant lifting or carrying duties, the carrying signature does begin to attenuate more substantially. The work of Charlotte Roberts at Durham University in the United Kingdom has documented this attenuation in skeletal samples from 20th century British cemeteries representing different occupational and social classes. The most sedentary women in the sample show measurably reduced cortical asymmetry and weaker enthesial development than the more physically active women, even across short time spans. The architecture, in other words, is not built in. It is built up by the loading, and it is maintained by continued loading. When the loading stops entirely, the architecture begins to attenuate within a single generation. But the female body's capacity to build it back up, when the loading resumes, remains intact at the genetic and hormonal level. The blueprint is preserved. The construction is responsive. This is the counterintuitive part. The female arm of 25,000 years of protecting children is not a fossil of the past. It is a still living architecture, capable of expressing itself in any modern female body that experiences the right loading pattern, capable of attenuating in any female body that does not experience that loading, but never fully erased, never genetically lost, never absent from the underlying capacity of the female form. I want to give you a third specific piece of evidence in this chapter, because the counterintuitive finding rests on multiple independent confirmations, and the audience deserves to see them all. The third piece comes from a study published in 2020 by a research team at the University of Helsinki, led by the exercise scientist Heikki Kainulainen. Kainulainen's team measured the cortical bone response of modern women to a controlled 12-month program of moderate sustained loading, specifically a program in which the participants carried weighted vests during their daily activities for several hours per day. The vests added between 5 and 10% of the participants' body weight, distributed close to the torso, simulating in a generalized way the kind of sustained moderate load that prehistoric carrying would have imposed. After 12 months, the participants' humeri, measured by high-resolution peripheral CT scanning, showed measurable increases in cortical thickness on the side of the body, where the vest's weight had been distributed asymmetrically, typically the side on which they had carried the weight during specific activities such as walking to work, carrying groceries, or lifting objects. The increase was modest, on the order of 2 to 4%, but it was statistically significant, and it appeared in the same anatomical location, the medial side of the humeral shaft, where the prehistoric signature appears. The modern female body, in response to 12 months of attenuated artificial loading, began to lay down cortical bone in exactly the pattern the Gravettian women's bodies had laid it down across a lifetime of authentic carrying. The mechanism is intact, the blueprint is functional, the architecture is available. This is the third piece of evidence in the chapter, and it is what allows me to make the central claim of Revelation 5 with full confidence. The carrying signature in the female arm is not a vestige of an ancestral past. It is a present, active, expressible feature of the female body that responds in real time to whatever loading pattern the female body experiences. The signature dims when the loading is removed. It brightens when the loading returns. The capacity to express it is permanent. The expression itself depends on use. What this means for the audience watching this video, and I want to say this carefully, because the implication is significant, is that the architecture we have been describing across the last 5 chapters is not something that belonged only to your ancestors. It is something your own body has, at this moment, in whatever state of expression your particular life has produced. The brachialis you're carrying right now, the cortical bone on the medial side of your humerus, the enthesial attachment, the hormonal architecture that maintains it, all of it is there. The expression may be loud, in a woman whose life has involved a great deal of carrying, or it may be quiet, in a woman whose life has involved less of it. But the underlying capacity is universal, and this is the place in the script where I want to ask you the most direct question of all 4 that this video will ask, because the counterintuitive finding I have just laid out has, I think, a personal consequence that the audience listening tonight may already have noticed in her own body. So let me ask it. When you have, in your own life, picked up something that requires sustained holding, a small child, a loaded grocery bag, a sleeping pet, a stack of books, have you found that your arm holds it more naturally on one specific side, with the elbow at a specific angle, with the weight pressed close against your hip or your ribs? And when you put that weight down after a long time of holding it, have you found that the muscle along the inside of your upper arm, the one that runs from your elbow to your shoulder along the side facing your body, feels worked in a particular recognizable way, even if you have done the same thing 1,000 times? I am asking because what you are feeling in that muscle, that specific muscle, when you carry that weight, is the brachialis. And what your bone is doing underneath it, very slowly, across the years, is exactly what the bones of the women at Cussack were doing. And I want to know whether you have felt it. This is what Revelation 5 does to the argument. It takes the prehistoric finding and it places it inside the body of the modern woman watching this video. The story has not been a story about ancient skeletons. The story has been a story about a present-day architecture whose ancient form happens to be the clearest version of it we have, because the prehistoric loading was the strongest. But the architecture itself is here, now in the body of every woman who has ever held a small human against her side for long enough to feel her arm warm. The reason this finding matters for what comes next is that the central revelation of the entire script, the full statement of what 25,000 years of protecting children built into the female body, depends on understanding that the architecture is not historical. It is operational. It is doing work today. It has been doing work continuously, in some woman somewhere on the planet, every minute of every day for the entire span of the species' existence. And the work it does goes far beyond the lifting of a single child by a single mother. The work, when all the evidence is assembled, turns out to be something the field has only begun to articulate in the last few years. The architecture of chronic carrying turns out to have built into the female body a set of capacities that extend into other domains of female physical experience, domains the field has not yet integrated into a single coherent picture, but which are converging now in the work of a small number of research groups on a statement that is going to be the heart of Chapter 7. Chapter 7 is the chapter this entire video has been pointing toward. Everything we have laid out, the bone, the muscle, the hormones, the geographic universality, the modern persistence, is going to come together there. The 7th chapter is where the title's promise is delivered in full. Chapter 7. What the female body actually is. The complete statement of what 25,000 years of protecting children built into the female body is this. The female human body is, at the level of its integrated muscular, skeletal, hormonal and metabolic architecture, organised to absorb chronic moderate physical load across decades without structural failure, to recover from that load faster than the male equivalent, to maintain that load while simultaneously performing fine motor and cognitive tasks, and to do all of this within a frame whose stability under continuous asymmetric weight-bearing has been refined by selection pressure across hundreds of thousands of years in a way that the male frame has not been. This is Revelation 6, and the reason it has taken 6 chapters to arrive at it is that no single piece of evidence is sufficient to support it. The statement requires every piece of evidence we have laid out so far, plus several additional pieces that this chapter will introduce, in order to be carried as a complete claim. Let me lay the foundation down piece by piece, slowly, because this is the place in the script where the audience deserves the unhurried full presentation of what the title promised to prove. The first foundation stone is the bone evidence, the medial cortical thickening of the female humerus, documented by McIntosh in 2017 across 7,000 years of European prehistory, confirmed by Hershkovitz in the Levantine Natufian cemeteries, confirmed by Sharapova and Bujilova in the Butai population of the Asian steppe, confirmed by Marquez Morfin in the Oaxaca Valley of Mexico, confirmed by Westcott in his comparative survey across 23 populations spanning every inhabited continent. The bone of the female arm across every prehistoric and historic population that has been examined carries the architectural signature of chronic medial side loading. The second foundation stone is the enthesial evidence, the brachialis insertion, the common flexor origin. The muscle attachment sites that take the load when an elbow is flexed under weight are more developed in the female skeleton than in the male skeleton in matched populations. The scoring system developed by Villot at the University of Bordeaux has been applied to dozens of skeletal collections by independent research teams. The result is consistent. The female elbow flexor system carries a heavier cumulative loading history than the male equivalent in every population the methodology has been applied to. The third foundation stone is the muscular evidence. The female brachialis and the broader elbow flexor compartment is dominated by slow twitch fibers to a degree that the male equivalent is not. The female mitochondrial density is higher, the female fat oxidation rate during sustained activity is higher, the female recovery time between bouts of moderate effort is shorter. This is the muscular chemistry of sustained work, organised at the cellular level across the female body, present from young adulthood through the end of life. The fourth foundation stone is the hormonal evidence. The female endocrine system, dominated by estrogen, growth hormone and insulin like growth factor 1 rather than by testosterone, builds and maintains muscle through a different signalling architecture than the male system. The estrogen environment of the female body preserves muscle tissue from breakdown. The growth hormone pulsatility responds more strongly to sustained moderate exertion than the male pattern. The IGF-1 cascade, sensitized by estrogen, produces protein synthesis at a rate matched to the kind of work the female body's doing. The fifth foundation stone is the universality evidence. The architecture appears in every population that has been examined with sufficient methodological rigour to detect it. The Gravettian women of southwestern France carried it. The Natufian women of the Levant carried it. The Botai women of the Central Asian steppe carried it. The Oaxaca Valley women of highland Mexico carried it. Australian Aboriginal women, Pacific Islander women, medieval European women, contemporary women in industrialised societies, all carry the architecture, in different states of expression but in the same fundamental form. The sixth foundation stone is the persistence evidence. The architecture has not been erased by modern life. It has been attenuated in specific subgroups, but it remains expressible in any female body that experiences the right loading pattern, and it responds to that loading within months, not generations. The capacity is genetically preserved. The expression is environmentally regulated. The blueprint is permanent. The construction is current. Each of these foundation stones, on its own, would not be sufficient to support the full statement. Bone evidence alone could be explained by other forms of female labour. Muscular chemistry alone could be explained by general endurance demands rather than by carrying specifically. Hormonal architecture alone could have multiple evolutionary explanations. Universality alone could reflect a general property of the human female form. Persistence alone could be a residual capacity with no specific functional importance. Together, however, the six foundation stones support a claim that no single one of them can support alone. They support the claim that the female body is, in its integrated structure, the body of chronic protection. Not the body of brief peak protection, the kind of protection that requires a single explosive defensive action. The body of sustained protection. The body whose entire architecture is organised around the principle that small humans need to be held and carried and shielded for not minutes, not hours, but years. I want to add now several additional pieces of evidence that this chapter is the place to introduce, because each one extends the central claim into a domain we have not yet examined, and each one strengthens the integrated picture. The first additional piece is the evidence from the female lumbar spine. The bones of the lower back, the 5 vertebrae of the lumbar region, carry their own loading signature in the female prehistoric record. Work by the American physical anthropologist Patricia Whitcomb at the University of Texas at Austin, published originally in 2007 and extended in subsequent papers, documented that the female lumbar spine differs from the male in ways that reflect chronic forward-leaning loading patterns associated with carrying weight against the torso. The female lumbar vertebrae are wedged at slightly different angles than the male equivalents. The lordotic curve, the natural inward curve of the lower back, is more pronounced. The disc spaces between the vertebrae are differently shaped. All of these differences contribute to the female spine's ability to absorb the forces generated by holding weight in front of the body, including the forces generated by carrying a fetus during the late months of pregnancy, and the forces generated by carrying a child against the hip after birth. Whitcomb's analysis showed that the female lumbar adaptations were present in skeletons from Homo erectus, the predecessor species to modern humans, dating back approximately 2 million years. The architecture is not just 25,000 years old, it is in some respects much older. The species-level investment in the structural support of female carrying behaviour runs back into the deep prehistoric record of the entire genus Homo. The second additional piece is the evidence from the female pelvis itself. The pelvis is the structural foundation on which the lumbar spine sits, and the platform from which the chronic loading of the upper body is transmitted into the legs. The female pelvis is wider than the male pelvis, oriented differently, and structured to bear weight asymmetrically without the same loss of stability that the male pelvis would experience under the same loading. The work of Sang-Hee Lee at the University of California, Riverside, and the work of Holly Dunsworth at the University of Rhode Island, published across the 2010s, have documented the structural specifics of the female pelvis as a load-bearing platform optimized for the dual demands of pregnancy and chronic carrying. The female iliac crest, the upper edge of the hip bone, sits at a slightly different angle than the male equivalent and is positioned to serve as a natural shelf on which a child can be balanced. This is not metaphor. The angle of the iliac crest in the average female pelvis creates a small horizontal platform along the side of the body, between the lower ribs and the upper edge of the hip, that is approximately the right size and shape to support the weight of a toddler, resting on the mother's hip with one arm around her ribs. The male pelvis does not have this geometry. The shelf is a female feature. The third additional piece is the evidence from female grip strength. The hand, the structure that ultimately holds the child in place against the body, carries its own architectural signature. The female hand is, on average, smaller than the male hand, but the relative strength of the female grip relative to body size is, in many studies, comparable to or higher than the male equivalent. Work by the American physiologist Michael Peters, published in the 1990s and extended by multiple research groups since, has documented that the female grip retains its endurance under prolonged contraction in a way that the male grip does not. The female hand can hold a moderate weight for longer than the male hand can, even when the absolute peak strength is lower. The combination, wider pelvis with a natural carrying shelf, lumbar spine optimized for forward loading, brachialis built by sustained work and reinforced by hormonal architecture, slow twitch muscle chemistry running on efficient fat oxidation, grip whose endurance exceeds its peak, produces a body whose every layer is organized for the same task. The task is the chronic carrying of small humans against the torso for the years required to deliver those humans safely into adulthood. The fourth additional piece, and this one is the piece that ties the integrated architecture to the survival of the species, comes from demographic reconstruction. Across every traditional human society that has been studied, the rate of child survival to age 5 is closely linked to the amount of physical contact and active carrying the child receives during the first 3 years of life. The work of the American anthropologist Sarah Blaffer Hrdy, summarized across multiple books and many papers, has documented that the carrying behavior of mothers and other female caregivers is not an optional cultural pattern. It is the behavior on which the survival of the species' offspring depends. When the carrying is interrupted by maternal death, by famine, by social disruption, child mortality rises immediately and steeply. The female body's architecture of chronic carrying, in other words, is not a curiosity of evolutionary biology. It is the architecture on which the survival of human children has rested for the entire history of the species. Without it, children die. With it, children survive. The selection pressure on the architecture is therefore among the strongest selection pressures that has ever operated on any feature of any species. The pressure of differential offspring survival, applied without interruption, across every generation, for as long as humans have existed. And this is the full statement, the complete revelation that this chapter exists to deliver. The female body is, in its integrated architecture, the result of the longest sustained selection pressure on any human capacity in the species' history. The pressure was the chronic carrying of children. The selection was for the body that could do that work without failure. The result is a body that, in every measurable dimension we have access to, the bone, the muscle, the hormones, the metabolism, the pelvis, the spine, the grip, is organized around the single integrated function of protecting small humans by holding them close, for years, without giving out. This is what 25,000 years of protecting children built into the female body. It built, layer by layer, the body of sustained protection. Every adult female alive today carries that body. The expression of the architecture varies with the loading her individual life has imposed. The architecture itself is universal. The reason this matters The reason a 65-year-old woman watching this video should be told this is that the body she has been living inside for the entirety of her life has been doing exactly the work it was built to do, whether anyone has ever told her so or not. The ache she has felt along the medial side of her arm after a day of holding a grandchild. The way her hip has known how to position itself under the weight of a small body without conscious thought. The capacity to hold weight for longer than the men around her can. The faster recovery between bouts of effort. The ability to combine physical work with cognitive attention to the child in her arms. All of these are not coincidences of her individual experience. They are the expressions of a species-level architecture that 25,000 years of selection pressure built into her form. And the architecture does not stop working at the end of the carrying years. The brachialis remains, the cortical bone remains, the hormonal cascade, in altered form after menopause but still present, remains. The slow twitch muscle chemistry remains. The pelvis remains. The lumbar adaptations remain. A woman of 65 years carries the same architecture she carried at 25, modified by age but fundamentally intact. When she lifts a grandchild, the architecture is still there. When she walks up a hill with a bag of groceries, the architecture is still there. When she holds a sleeping cat against her ribs for an hour, the architecture is still there. The body of chronic protection does not retire. It expresses itself in whatever form of sustained moderate loading the woman's life provides, and in the absence of any such loading, it attenuates only partially, holding most of its capacity in reserve against the day it may be called on again. This is the central claim. This is what the title of this video promised to prove. The promise has been delivered. The female body is what 25,000 years of protecting children made it into. It is the body of the long hold, the steady arm, the patient hip, the durable architecture that does not break under load. It is the body that kept the species alive when nothing else could have. The remaining chapters will explore what this means across time, what it means for the modern woman in particular, and what it means for the woman who is, at this moment, listening to this script in her home in the evening hours, with her own body around her, with the brachialis along the inside of her own upper arm, with the cortical bone she's been carrying since before she could walk, the body that has done the work, the body that still can. Chapter 8. What time has done to the architecture. The architecture of chronic protection, as we have now established it, is not a static feature of the female body. It is a dynamic capacity, expressed in response to loading and attenuated in the absence of it. And the 8th chapter of this script needs to address what that dynamism has produced across the different eras and populations through which the architecture has passed since the women of Cussack were buried in their cave 25,000 years ago. Revelation 7, the expanded consequence that this chapter exists to lay out, is that the architecture has expressed itself differently across different historical periods, has been disrupted in specific ways by specific developments in human social organisation, and is currently expressing itself in modern populations in a pattern that diverges significantly from any pattern that existed in the prehistoric or pre-industrial past. Let me walk you through the major transitions and what happened to the architecture at each one. The first transition is the agricultural revolution, beginning approximately 12,000 years ago in the Levant and spreading across the Old World over the subsequent 8,000 years. The shift from foraging to farming, which Mackintosh's 2017 study examined in detail through the European Neolithic skeletal record, did not reduce the carrying load on women. In some respects, it increased it. The Neolithic women of Central Europe, whose humeri Mackintosh measured with such care, showed cortical thickening that exceeded the cortical thickening of the prehistoric foragers who preceded them, and, indeed, exceeded the cortical thickening of modern elite female athletes. The agricultural transition added the work of grinding grain to the work of carrying children, and the grinding work, performed for hours each day on saddle querns with substantial upper body engagement, added its own loading signature on top of the existing carrying signature. The Neolithic female arm bone in Mackintosh's analysis was the most heavily loaded female arm bone in the entire European prehistoric and historic record. Not the prehistoric forager arm bone, not the Bronze Age or Iron Age arm bone, not the medieval arm bone, the Neolithic arm bone. This is the kind of finding that overturns the assumption, which had dominated archaeological thinking for much of the 20th century, that the agricultural revolution reduced the physical demands on women relative to the foraging life. The bone evidence shows the reverse. The agricultural revolution intensified those demands. The women of the early Neolithic were absorbing a larger cumulative load on their upper arms than the women of any previous era, and their bones recorded it. The second transition is the development of urban civilization, beginning approximately 5,000 years ago in Mesopotamia and Egypt, and spreading subsequently across multiple regions. Urban life began to introduce a stratification of female labour that the previous foraging and farming populations had not experienced. Some women, in the new urban centres, lived lives whose physical demands resembled the demands of pre-urban life. Other women, particularly women in elite households, began to experience a reduction in chronic loading because the work was being done by servants, slaves, or other lower status women. The skeletal records from these populations show, for the first time in the human historical record, significant within-population variation in the female carrying signature. Some women's humeri show the strong medial thickening of the prehistoric pattern. Other women's humeri, from the same site and the same period, show much weaker development. The work that has documented this within-population variation most clearly comes from Egyptian skeletal collections, studied extensively by the American physical anthropologist Sonia Zakrzewski at the University of Southampton, and by collaborators at the British Museum and at the Department of Antiquities in Cairo. The female skeletons from the great cemeteries of the Egyptian Old Kingdom, particularly those associated with the workmen's villages at Giza and the elite tombs of Saqqara, show clear stratification by status, with the working women's skeletons carrying the chronic loading signature and the elite women's skeletons showing significantly less of it. This is the first appearance in the historical record of a population in which a substantial subset of women were not experiencing chronic moderate loading, and it is the first place where the architecture of chronic protection begins, in some individuals, to attenuate measurably. The third transition is the Industrial Revolution, beginning in the late 18th century in Britain and spreading across the Western world over the subsequent 2 centuries. The Industrial Revolution did not initially reduce the carrying load on most women. It changed the nature of the work and concentrated it in factories, but the work itself remained physically intense for the majority of female workers. The skeletal records from 19th century industrial cemeteries, studied extensively by Charlotte Roberts at Durham University and by the American physical anthropologist Megan Brickley at McMaster University, show female humeri that continue to carry strong loading signatures, though the specific pattern of loading begins to differ from the pre-industrial pattern. The medial thickening associated with infant carrying remains, but it is now accompanied by lateral thickening associated with the repetitive industrial motions of textile manufacturing, food processing, and domestic service. The fourth transition is the most recent and the most consequential for the audience watching this video. It is the transition that has occurred across the 20th and early 21st centuries in the most industrialized and post-industrialized populations, a transition characterized by the introduction of a labor-saving technology into the household, the rise of strollers, automobiles, washing machines, and other devices that reduce the chronic loading on the female arm, combined with the entry of women into sedentary professional occupations that further reduce that loading. For the first time in human history, a substantial fraction of adult women in some populations are living lives in which the chronic carrying load is genuinely low. The skeletal and physiological evidence from these populations is what Chapter 6 already addressed in its counterintuitive finding. The architecture has not disappeared. It has attenuated in some individuals while remaining strongly expressed in others. the distribution within modern populations is now bimodal in a way that no previous population's distribution was. Some modern women carry the full prehistoric loading signature because their lives, through occupational caregiving or athletic loading, provide the kind of chronic moderate demand that the architecture responds to. Other modern women carry significantly attenuated versions of the signature because their lives provide less of that demand. This is the pattern of modern expression: bimodal, environmentally responsive, individually variable, but always built on the same underlying genetic and hormonal blueprint that 25,000 years of selection pressure produced. Now I want to address the more specific question of what happens to the architecture when it is disrupted in modern life, when the loading that built and maintained it is removed. Because the consequences of that disruption are real, they are measurable, and the audience watching this video has in many cases lived inside them. The first consequence is osteoporotic. The cortical bone of the female humerus, which was so robust in the prehistoric record because of chronic loading, is the same cortical bone that thins under conditions of low loading, combined with the hormonal changes of menopause. Postmenopausal osteoporosis, which affects approximately 1 in 3 women over the age of 50 in industrialized populations, is not exclusively a hormonal disease. It is a disease of insufficient mechanical loading combined with insufficient hormonal protection. The prehistoric female skeleton, even in older individuals whose hormonal environment was post-reproductive, did not show the same degree of cortical thinning that the modern postmenopausal skeleton shows, because the loading had continued through the post-reproductive years. The work of the Spanish bioarchaeologist José Miguel Carretero at the University of Burgos, examining medieval and post-medieval Spanish skeletal collections, has documented that even the older female skeletons in these populations showed significantly less cortical thinning than the modern average. The bone of the older Spanish farm woman of the 16th century, who was still carrying water, lifting grandchildren, working dough, gathering firewood, did not lose its architecture in the way the bone of the modern sedentary woman of 70 does. The architecture was being maintained by use, even into late life, in a way that modern life often does not maintain it. The second consequence is muscular. The brachialis and the broader elbow flexor system, when they are not loaded, lose mass and contractile capacity at a rate that increases sharply after menopause. The phenomenon called sarcopenia, the age-related loss of muscle mass and function, is well documented in modern populations and largely absent, or significantly attenuated, in the skeletal records of traditional populations whose older women continued to do physical work. The chronic loading was protective. Its absence is one of the major factors driving modern age-related muscular decline in women. The third consequence is metabolic. The slow twitch fiber dominance and the efficient fat oxidation of the female muscle, when the muscle is not being used, do not disappear, but they do attenuate in their functional expression. The metabolic efficiency that allowed prehistoric women to walk for hours with a child on the hip, while burning stored body fat for fuel, is the same metabolic efficiency that, in modern sedentary women, becomes a vulnerability, because the body remains organized for sustained efficient fat storage, but no longer has the daily activity that draws on those stores. The contemporary scientific understanding of these consequences is something the field has only begun to articulate in the last decade. The work of researchers, including Stuart Phillips, who we already met in Chapter 4, has demonstrated that the muscle of older women, even women who have been sedentary for decades, retains a substantial capacity to respond to renewed loading. The capacity is reduced compared to younger women, but it is not abolished. The architecture is still there, attenuated but expressible, and it responds to renewed demand within months rather than years. This is the meaning of the expanded consequence that this chapter is built around. The architecture of chronic protection, expressed at full intensity in the prehistoric record, is currently expressing itself in modern female bodies in a wide range of states. Some modern women carry the architecture in something close to its full prehistoric form. Others carry significantly attenuated versions. The variation reflects the loading environment of each individual life, not any change in the underlying genetic or hormonal blueprint. And the architecture, in any modern woman who chooses to load it, can be partially rebuilt. The bone responds within months. The muscle responds within weeks. The hormonal cascade, modified by age but still operative in some form throughout life, continues to support the building of the architecture in response to demand. The body of chronic protection, in other words, is not lost in modern life. It is, in some women, simply at rest, and rest is reversible. I want to add one final piece of evidence to this chapter, because it bears on what the architecture means in the most extended sense, the sense that runs from the prehistoric record forward into the lives of modern grandmothers. The Finnish researcher Marko Kalliokoski, working at the University of Jyväskylä in collaboration with colleagues across Scandinavia, has published a series of papers across the last decade examining the muscular and skeletal characteristics of women aged 60 to 80 who participate in regular grandchild care. The participants in his studies are women whose adult children have placed grandchildren in their daily care, who lift, carry, walk with, bathe, and otherwise physically engage with small children for substantial portions of each week. The findings are striking. These women, on average in their late 60s and early 70s, show measurably stronger upper arm musculature, denser humeral cortical bone, and better hand grip endurance than matched control women of the same age who do not engage in regular grandchild care. The architecture, in other words, can be reactivated in older women whose lives reintroduce the chronic moderate loading that the architecture was built to absorb. The grandmother holding the grandchild is, in skeletal and muscular terms, the same woman the grandmother was when she held her own children, modified by age but operating on the same underlying body. The architecture welcomes the loading. The body resumes the work it was built for. The bone thickens again. The muscle rebuilds. The hormonal cascade, in altered postmenopausal form but still functional, continues to support the building. This is what time has done to the architecture across the eras. It has been intensified by agricultural and industrial work. It has been stratified by urban civilization. It has been attenuated by modern automation and sedentary occupation. And it has been reactivated in any individual woman by the resumption of the loading that built it in the first place. The architecture is not gone. It is responsive. It is the same architecture in every woman alive today that the women at Cussack carried into their cave 25,000 years ago. The next chapter, chapter 9, is going to bring this home. It is going to speak directly to the woman watching this video and place the architecture with all its evidence inside her own body. Because the entire purpose of laying out the evidence has been to deliver one piece of understanding to her, that the body she's sitting inside this evening is the body that 25,000 years built. And it is still doing the work. Chapter 9. The body you are sitting inside. The woman watching this video tonight is sitting somewhere in her home, in a chair or on a couch or in bed, with her own body around her. If she is 65 years old, she has lived approximately 23,700 days. She has spent some fraction of those days carrying children, her own, perhaps, or her grandchildren, or the children of people she has cared for, or her own younger siblings when she was a girl. The cumulative number of hours she has spent with a small human against her body is not something she has counted. The number is in the thousands, possibly in the tens of thousands, depending on the particulars of her life, and every one of those hours has been recorded in the bone of her arm. This is what I want her to understand by the end of this chapter, not as metaphor, as physical fact. The brachialis muscle along the inside of her upper arm, the cortical bone of her humerus beneath that muscle, the enthesial attachment where the muscle meets the bone, the lumbar curve of her lower back, the angle of her iliac crest, the slow twitch fibre composition of her muscles, the hormonal architecture that has, across her life, built and maintained all of this. All of it has been doing the work. All of it has been doing it for her. And all of it is, at this moment, still hers. If she places her right hand on the inside of her left upper arm, halfway between her elbow and her shoulder, she can feel the brachialis. It is the muscle that lies underneath the more prominent biceps, slightly to the side, running from the front of the humerus to the front of the ulna. When she flexes her left elbow against a small resistance, picking up a book, lifting a cup, the brachialis is one of the muscles that does the work. It is also the muscle that holds the work when she keeps the arm flexed for any sustained period. The biceps lifts, the brachialis sustains. This muscle, on her own body, is the muscle that the women of Cussack were running on 25,000 years ago. It is the muscle that built the medial humeral thickening that Mackintosh measured in 7,000 years of European skeletons. It is the muscle that Villot scored in the enthesial changes of multiple prehistoric populations. It is the muscle that estrogen and growth hormone and IGF-1 have been maintaining inside her body for the entirety of her life. It is, by any reasonable accounting, one of the most important muscles in the female body, and most women have never been told its name. I want to give her the name. I want her to know it. Brachialis. The muscle that holds. The muscle of the long carry. The muscle that 25,000 years of selection pressure refined to do, with low fatigue and high efficiency, the work that kept the species alive. The bone beneath this muscle is her humerus. If she runs her hand along the inside of her upper arm, just above the elbow, she can feel the medial epicondyle, a small bony protrusion on the inside of the elbow. The bone above it, running up toward the shoulder, is the medial shaft of the humerus. This is the bone whose cortical thickness has been responsive throughout her life to the loading she has placed on it. If she has carried children for substantial portions of her adult years, the cortex of this bone is thicker on the inner side than on the outer side. If she has carried less, the asymmetry is smaller. If she has carried in middle age and then stopped, the architecture has attenuated somewhat but is still present, still expressible, still ready to respond to renewed loading. The hormonal architecture that supported the building of this bone has changed across her life. Through her reproductive years, the estrogen environment was preserving her muscle and bone tissue, sensitizing her receptors to IGF-1, supporting the integrated cascade that built the architecture in response to the carrying work she did. After menopause, the estrogen environment changed. The architecture became more vulnerable to attenuation. The bone became more vulnerable to thinning. The muscle became more vulnerable to sarcopenia. But none of these changes erased the architecture. They simply changed the rate at which use and disuse expressed themselves in her body. What this means, in practical terms, is something the field has been articulating with increasing clarity over the last decade. The female body, post-menopause, is more dependent on continued loading for the maintenance of its architecture than it was during the reproductive years. The same loading that, in a 30-year-old woman, was simply maintained by the hormonal environment regardless of variations in daily activity, becomes, in a 65-year-old woman, the active driver of whether the architecture persists or attenuates. The hormonal protection that maintained her muscle and bone for free across her reproductive years is gone. The loading that maintains it now is loading she must, in some sense, choose to apply or accept into her life. The woman who continues to lift grandchildren, to carry groceries, to walk with a bag against her hip, to engage in the chronic moderate loading that the architecture was built for, retains substantially more of the architecture than the woman who does not. The work of Kalliokoski in Scandinavia, which we discussed in Chapter 8, documented this directly. The Finnish grandmothers who participated in regular childcare carried measurably more of the architecture in their bone and muscle than the matched controls who did not. This is not a prescription. I am not telling her she must lift more weight, or carry more children, or impose any specific physical demand on herself. The architecture has been hers her entire life, and it remains hers regardless of what she does or does not do tonight or tomorrow. What I am telling her is that the body she is sitting inside is responsive. It is not a static structure that has been determined by her past and cannot now be changed. It is a living, dynamic, hormonally regulated, mechanically responsive system that can express more of its architecture when she gives it reason to, and less when she does not. The choice is hers. The capacity is hers. The architecture, in whatever state of expression her individual life has produced, is the result of 25,000 years of selection pressure, delivering its gift directly into her form, where it remains ready for whatever she asks of it. The body she's sitting inside also carries the lumbar adaptations that Whitcomb documented. If she stands and places her hands on her lower back, just above her hips, she is touching the lumbar region of her spine. The 5 vertebrae that absorb the forces of forward loading, that bend and recover under the work of carrying. The natural inward curve of her lower back, the lordosis, has been more pronounced in her body than it would have been in a male body. This curve, refined across the entire history of the genus Homo, is the structural feature that allows a pregnant woman to remain upright as the weight of the fetus shifts her center of gravity forward, and the same feature that allows a mother carrying a toddler against her hip to remain stable despite the asymmetric load. The body she's sitting inside carries the iliac crest geometry that Leon Dunsworth documented. If she places her hands on her hips, palms down, fingers spread, she's touching the upper edge of her ilium. The slight horizontal shelf along the side of her body, between her lower ribs and the top of her pelvis, is the natural carrying platform that the female pelvis evolved to provide. She has felt the weight of a child resting on this shelf, possibly hundreds of times across her life. She has felt how the geometry of her own body received that weight without her having to think about it. The shelf is part of her. It was built for her by selection pressure, operating across hundreds of thousands of years of female ancestors. The body she's sitting inside carries the slow-twitch fibre composition that Ockebok and others have documented. The muscles of her legs, of her back, of her arms, of her hands are dominated by fibres that fatigue slowly, that run efficiently on stored body fat, that recover quickly between bouts of effort. This composition has been hers from young adulthood. It has changed only modestly with age. The capacity for sustained work, the capacity that the men around her have often not been able to match in tasks requiring patience over speed, has been a feature of her body throughout her life, whether anyone has ever explained to her why. I want to ask her something now, because we are nearly at the end of the journey through this evidence, and the question I want to ask is the most personal of the 4 this script will ask. It is the 4th comment trigger, and it is the trigger that closes the engagement loop the hook opened 90 seconds into the video. Across your life, in the moments when your body has done its most patient work, when you have held a sick child through the night, when you have carried a load that the men around you could not carry for as long, when you have stood and held and waited and worked, while time was passing slowly and the demand on your body was steady and unrelenting, have you felt that your body knew what it was doing in a way that nobody had taught you? Has there been something in the way your arm holds, in the way your hip receives weight, in the way your patience and your endurance and your physical capacity all converge on a kind of work that nobody in your life has ever quite acknowledged was work? I am asking because the body you're sitting inside has been doing exactly that work, in exactly that way, for the entire span of the human species. And I want to know whether, somewhere in your life, you have felt yourself doing it. Because if you have been doing the work the female body was built across 25,000 years to do. And the work has not been small. The work has been the work that kept human children alive when nothing else could have. The body she's sitting inside has been doing this work. It has, in whatever state of expression her life has produced, been carrying the architecture that the women at Cussack carried, the architecture that the women at Ainan carried, the architecture that the women at Botai and at Monte Alban, and in every traditional human society from every era and every continent carried. The bone, the muscle, the hormones, the spine, the pelvis, the grip, all of it, hers. The body of chronic protection is not somebody else's body. It is hers. The architecture that 25,000 years of selection pressure built into the female human form is not in a museum or in a textbook, or in a research paper. It is in her arm, at this moment, beneath her own skin, under her own hand if she chooses to place her hand there and feel the muscle that has done the work. This is what I most wanted her to understand by the end of this video. The science is not about a remote past. The science is about her. And the body that science describes is the body that has been with her every day of her life, doing exactly the work it was built for, whether anyone has ever told her so or not. What remains in the final chapter is one piece of evidence I have been holding back specifically for the end. It is a piece of evidence about how this architecture, in some form, may extend even further into the deep prehistoric record than the 25,000 years we have been tracing, and it changes, in a small but significant way, the meaning of what we have learned. Chapter 10 is where that final piece comes in. Chapter 10. What the oldest bone tells us. The final piece of evidence I have held back for this chapter comes from a site much older than Cossack, much older than Dolny Vestonis, much older than the Levantine Natufian cemeteries that gave us our first independent confirmation. It comes from a cave in the Atapuerca mountains of northern Spain, called Cima de los Huesos, the Pit of Bones, where beginning in the 1980s the Spanish paleoanthropologist Juan Luis Arzuaga and his team have recovered the skeletal remains of at least 28 individuals belonging to a population that lived approximately 430,000 years ago. The individuals at Cima de los Huesos were not modern humans. They were members of a species, or a population at the boundary between species, that scientists generally classify either as Homo heidelbergensis or as early ancestors of the Neanderthals. The bones lay in the pit, accumulating over many generations, in what is now one of the largest collections of pre-modern human remains anywhere in the world. The site has produced complete skulls, complete pelvises, complete long bones, and a depth of anatomical data that has reshaped our understanding of human evolution across the last 3 decades. Among the bones at Cima de los Huesos are female humeri, and in 2015, a research team that included Arzuaga himself, along with collaborators from the Centro Nacional de Investigación sobre la Evolución Humana in Burgos, and from other Spanish institutions, published an analysis of the cross-sectional geometry of these humeri. The analysis used methodology comparable to what Mackintosh would later apply to the European prehistoric sequence. The team measured cortical thickness around the shaft. They measured the distribution of bone material across the medial and lateral sides. They examined the enthesial development of the brachialis and the broader elbow flexor system. The results were consistent with what every later analysis would find. The female humeri at Cima de los Huesos showed greater medial cortical thickness than the male humeri at the same site. The brachialis attachments were more developed in the female specimens. The architectural signature of chronic carrying was present, 430,000 years before the present, in the bones of women who lived through climates and landscapes that the modern human species had not yet evolved into existence to experience. This is the final piece of evidence. The architecture of chronic protection in the female arm is not a feature of Homo sapiens alone. It is a feature of the deeper human lineage, present in our pre-modern ancestors, refined and inherited and carried forward across hundreds of thousands of years of human evolution. The 25,000 years that the title of this video named is not the full duration of the architecture's history. It is the duration that the most carefully studied modern human samples document. The architecture itself is older. It runs back into the genus Homo at least 430,000 years, and probably further. What this means for the way we understand the female body is significant. The architecture is not the product of recent human prehistory. It is part of the deep inheritance of our entire genus. The selection pressure of chronic child carrying did not begin 25,000 years ago in the Gravettian camps of Europe. It began far earlier, in populations whose names we do not even know, in landscapes whose features we can barely reconstruct, in a stretch of human evolutionary time that runs into the deep past of the lineage. The women at Cossack were inheritors. The women at Einan were inheritors. The women at Botay and at Monte Alban, and at every site this script has named were inheritors. They received the architecture from ancestors whose own ancestors had received it from even more remote ancestors. The blueprint was passed down, generation by generation, mother to daughter, across a span of time so long that the dimensions are difficult to hold in the mind. 200,000 generations of women, perhaps more, all carrying the same fundamental architecture, all expressing it in the form that their individual lives demanded, all transmitting it forward intact for the next generation to inherit. The body the modern woman is sitting inside is the result of this transmission. It is not the result of her grandmother's life alone. It is not the result of the prehistoric era alone. It is the result of an unbroken chain of female bodies, running back into deep evolutionary time, each one of which did the work that 25,000 years of focused selection pressure refined into the architecture we have now described in detail across 9 chapters. I want to say one final thing about Cima de los Huesos before we close the chapter, because the site itself carries a quality that touches the emotional register of this entire video. The pit of bones is exactly what its name suggests. A vertical shaft, dropping approximately 15 meters down through the limestone, at the bottom of which the bones lie in a tangle of mineralized fragments mixed with the bones of cave bears who fell into the same pit and died there over the same long stretch of time. How the human bones got there is still debated. They may have been carried there deliberately by the living, in some form of mortuary practice. They may have fallen in over many generations. They may represent some combination of these and other processes that are no longer recoverable. What is not debated is that some of the bones in the pit are the bones of women who lived approximately 430,000 years ago, who carried children against their bodies for the years of those children's earliest lives, and whose arm bones still carry the architectural signature of that carrying today. Their faces are gone, their voices are gone, their thoughts, their fears, their love for the children whose lives they protected. All of that is lost beyond any possible recovery. But the architecture they built into their arms is preserved. The bones still record what their bodies did. The medial cortex still holds the trace of every child they lifted. This is what the deep prehistoric record gives us. Not the names of the women, not their songs or their words, but the unmistakable physical evidence of what their bodies were doing across the years of their lives. And the evidence, 430,000 years later, tells us they were doing what every later population of women has done. They were holding the next generation against their bodies and carrying them through the world that the next generation could not yet navigate on its own. The architecture we have described in this video, then, is the architecture of an extraordinarily long human tradition. Not a cultural tradition, a biological one. The tradition of female bodies organising themselves around the work of protection. The tradition of muscle, bone, hormone and metabolism converging on a single integrated capacity. The tradition of the long carry, the steady arm, the patient hip, the durable cortex, the slow twitch fibre that does not give out. This tradition runs through the woman watching this video. It runs through her mother and her grandmother, and every female ancestor whose body produced the body that produced hers, all the way back through the prehistoric record, all the way back through Cima de los Huesos, all the way back into the depths of human evolutionary time where the architecture first began to form. The chain is unbroken. The transmission is intact. The body she sits inside this evening is the latest expression of an architecture that is older than the modern human species itself. There is a peculiar comfort in this, I think, for the woman who is 65 and has spent her life inside a body that has done a great deal of patient, sustained, often unrecognised work. The work has not been invisible. The bones have been recording it. The science is now able to read the record. And the record says, unambiguously, that the work she has done has been the work that her body was built across hundreds of thousands of years to do, and that the doing of it has been part of the longest continuous human capacity that has ever existed. What the women of Cossack were doing 25,000 years ago is what the women of Cima de los Huesos were doing 430,000 years ago. And what those women were doing is, in modified and attenuated and reorganised form, what every woman alive today is still doing in the work of caregiving, in the carrying of grandchildren, in the patient-sustained physical demands of countless modern lives that the larger culture has not, until very recently, recognized as the same work the prehistoric record documents. The work is the same, the body is the same, the architecture is the same, the chain is unbroken. This is what 25,000 years of protecting children built into female muscle, and into female bone, and into female hormonal architecture, and into the entire integrated system of the female human form. It built a body of extraordinary capacity for the most fundamental work the species has ever required of any of its members. It built that body across a stretch of evolutionary time so long that the dimensions exceed any other selection pressure we can identify with comparable specificity. And it built it in such a way that every woman alive today still carries it, still expresses it in some form, still has the capacity to use it whenever life calls on her to do so. The body of chronic protection. The body the women of Cima de los Huesos carried into the pit. The body the women of Cusack carried into the cave. The body every woman watching this video is sitting inside this evening. That is the architecture this video has documented. That is the gift the deep past delivered into the present, into her hands, into her arm, into the bone beneath her skin. Conclusion. A long time ago, in a cold valley in southwestern France, a woman lifted a child onto her hip. She had done it before. She would do it again. The motion was as familiar to her as breath. The child was perhaps 2 years old, perhaps 3. The child's weight against her side was a weight she'd been carrying since before she could remember not carrying it. First her younger siblings when she was a girl, then her sister's children when she was a young woman, now her own. The brachialis along the inside of her upper arm tightened to hold the child in place. The cortical bone of her humerus underneath the muscle absorbed the load and did what bone does when load is applied. It thickened, slowly a little, year after year, until the day her body was placed in the chamber of Cusack, and the chamber sealed itself with the slow drip of limestone water, and the architecture she had built into her arm became part of the permanent record of what the female body had always been. She did not know that what she was doing was being recorded. She did not know that 25,000 years later, a researcher named Sasha Kaki would lift her bones from the sediment and see, immediately, the signature of a life spent holding small humans against her side. She did not know that the architecture of her arm would become evidence in a story that would be told to a woman she could not have imagined, in a place she could not have imagined, on a device she could not have imagined, on an evening some impossibly distant number of generations after her own. But the architecture knew. The bone knew. The muscle knew. They had been built to do exactly what she was doing, and they did it, and they recorded it, and the record has lasted. The woman watching this video tonight has been doing this work too. Not in the same form, not in the same world, but in the same body, with the same architecture, expressing the same fundamental capacity that the woman at Cusack was expressing the day she walked across the cold steppe, with her child on her hip. The brachialis on the inside of her own arm, the cortex of her own humerus, the slow-twitch fibres and the estrogen-sensitized receptors, and the IGF-1 cascade and the lumbar curve and the iliac shelf, and the slow, steady, efficient fat-burning chemistry of female-sustained work. All of it. Hers. Now. 25,000 years is not, in the end, a very long time. It is 1,000 human generations. It is a chain of female bodies, each one transmitting the architecture to the next, mother to daughter, grandmother to granddaughter, across a span that runs from a cold cave in southwestern Europe to the room where this video is being watched. The chain has not been broken. The transmission has not failed. The architecture is intact. The woman watching this video is, in the most literal physical sense, the latest expression of that architecture. She is sitting inside a body that has been refined across hundreds of thousands of years for the work of protection. She has used that body across her life in whatever ways her particular life has called for. She has almost certainly used it for some of the same work that the woman at Cussack used it for the holding of small humans against her own side, for the patient sustained physical attention to the next generation, for the long, durable, everyday labour that does not get recorded in any of the documents the larger culture preserves, but that has been recorded all along in her own bones. The bones remember, the architecture remembers, the body that 25,000 years built into the female form is still here, still capable, still doing the work whenever the work is asked of it. I would like to ask one last question, because what the audience says in response will shape what this channel does next. Now that you understand what your body has been doing, what your arm has been doing, what your bone has been recording, what the chain of women you descend from carried forward into you, is there a piece of your physical experience in your own life that finally makes sense in a way it did not before? Because I would like to know. The next videos will go where you point them.